Hi and welcome to my OCR A A-Level Biology revision session with me, Christine. So today's lesson I want to look at selective reabsorption at the proximal convoluted tubule known as the PCT. So let's just remind ourselves about the structure of the kidney nephron. So we have our glomerulus and our Bowman's capsule where ultrafiltration will occur. The nephric filtrate that is then formed is going to then make its way through the proximal convoluted tubule, and that's where we're going to look at what happens. So now remember, these are all in the cortex part of the kidney structure. So when we look at the proximal convoluted tubule, we like to compare it with the distal convoluted tubule. And the first thing to note then is if you look, you can clearly see there's a brush border around the proximal convoluted tubule compared to the distal convoluted tubule. That is one distinguishing feature. The second distinguishing feature is that you can actually see that there's a smaller lumen in the proximal convoluted tubule compared to the distal convoluted tubule. So this is something that they may ask you to identify which one of the pictures is showing the proximal convoluted tubule compared to the distal convoluted tubule. So your key identifying feature is look for the lumen, and does it have a darker shaded brush part? If it does, then that is your proximal convoluted tubule. So let's just have a look then at what happens then in this proximal convoluted tubule. So the first thing to know is this is the site of selective reabsorption. Now remember, ultrafiltration meant that all the small molecules inside our blood plasma, due to the high hydrostatic pressure, were squeezed and forced out of the plasma into the glomerular filtrate. So if we look at our nephric filtrate, then what we can see is at the Bowman's capsule, there is 100% glucose has been moved across and squeezed into this nephric filtrate. Well, what we want to do is we want to get that glucose back into the bloodstream, because glucose is our respiratory substrate that is required for glycolysis. So we don't want to lose glucose in our urine. We don't want to excrete it out of the body. And that is a distinguishing feature. If somebody has diabetes, then glucose will be found in their urine. So we know that we've got our microvilli, and our microvilli are there to increase the surface area. So because we know that those microvilli are there to increase the surface area, remember microvilli, they'll be able to move. If they're able to move, that links to your cytoskeleton. So not only are they increasing the surface area by being present, but it also means that they're a membrane which is going to have proteins attached to it, and that's what we're going to look at now. The first thing we need to note is at the proximal convoluted tubule, active transport occurs. Now that is your sodium-potassium pump. So your sodium-potassium pump are going to pump sodium out of the proximal convoluted tubule cell and potassium in. So by pumping the sodium out of the cell, what's going to happen is there's going to be a concentration gradient. We are pumping, so therefore we're taking it from a low concentration to a high concentration, and we're wanting to maintain a very low concentration of sodium inside our proximal convoluted tubule cell. And what that will then mean is that facilitated diffusion can occur, and that can occur down a co-transporter. So what we have is our sodium concentration inside our proximal convoluted tubule cell is now lower than the sodium concentration in the filtrate. So sodium is going to diffuse down its concentration gradient. Remember, it's a charged ion, therefore it needs facilitated diffusion because it needs a helper. It will not get across that phospholipid bilayer. Well, the glucose can't make its way across either because it's a polar molecule. So again, what needs to happen is a protein needs to move them across that phospholipid bilayer. And that's done through a process called co-transportation, where the glucose and the sodium come into the proximal convoluted tubule cell together. So what we've actually got is that the concentration gradient that was created by our sodium potassium pumping the sodium out, ensured that the sodium was going to diffuse into the proximal convoluted tubule cell, and it would bring the glucose along with it. Well, now that glucose can diffuse from the proximal convoluted tubule cell into the bloodstream because the specific channel, the GLUT channel, is present in the membrane, allowing for that movement across in that direction. So it's important that we note that the potassium that was pumped into our proximal convoluted tubule cell is going to change the ion concentration inside that cell. So the potassium is able to diffuse through facilitated diffusion down its concentration gradient through either the channel or the carrier proteins that are specific for potassium. So this is where your knowledge of the membrane structure comes into play. For us to understand what's happening at the proximal convoluted tubule, we have to understand that it involves these transport of ions to get the glucose back into the bloodstream. And this continues all the way down through the proximal convoluted tubule, so that when you get to the end of the proximal convoluted tubule, all of the glucose should have been reabsorbed back into the bloodstream. So I hope you've liked this video, and if you have, then please do click on the like button and subscribe to my channel. Also, do check out my revision platform, www.aiqchat.com.